first time I've taken advantage of Mark, because several years ago, when we had the last venting meetings here in Mobile, Mark was talking in one particular room, and we were trying out these remote keyboards. Well, the person in the next room over using the remote keyboard kept hitting page down and changing Mark's slides for him. Thankfully, Mark and I have been friends a long time, so I got off the hook. But, so this is I just realized, I just remembered that. Oh, there's a pattern there. There is. You'll never come back again. So for those of you that don't know Dr. Butler, he's the eminent ecology at Old Dominion University, located in Norfolk, Virginia. He's a graduate of Florida State.
Go Knowles!
And he's going to be counseling Jameis Winston here in the next few weeks.
That's right.
The playoff things.
We did take crabs, so.
Yeah. I also add that he is not as old as our current graduate chairman of university programs from Florida State, who is still suffering from jet lag in Lower Australia. So that would be Ken Heck, for those of you that are wondering. Mark's interests in ecology are very broad. He apparently brought some music with him. And so for those of you that get a chance to talk to him, you'll find plenty of common ground. His training is in freshwater and terrestrial, just like mine was in the early days, so he knows a lot about a whole bunch of stuff. I was fascinated by his title. He covers everything from societal pestilence to encounters with aliens from a lobster's perspective. You know, I assumed NASA was hiding that stuff, but apparently you're on the inside of this. The title of his talk is Lobster Tales, Home Pestilence, and Alien Encounters.
Thank you, John.
Good luck to you.
Yeah. Hopefully all the various and sundry technical glitches are over with. And I'm told I'm tethered to this place. I'm usually a pacer. No. To move the larvae around in the system. It also has a spatial component. So it has a GIS spatial component composed of over 3,000 sort of habitat polygons, reef habitat polygons around the Caribbean where these lobsters might begin and end up. Just looking a little bit closer, you can see here the polygons differ a little bit in shape, and they roughly map the existence of coral reef, major coral reef areas around the Caribbean. Now what we had to do, and it was quite actually useful to be working with lobsters in this case, you've got to start out in one of these biophysical models. If you're going to be looking at dispersal, the first question is, where are all the eggs starting? Now the problem with many reef fish is there's just really no way to know this. There's no really comprehensive stock assessments, but with lobsters being so important, there was reasonably good data for this. So in addition to looking at the literature, we actually sent out surveys and worked with managers throughout the Caribbean to get as best we could a good feel for the magnitude and seasonality of spawning at all these little nodes in the Caribbean. Now again, they were not perfect set of data, but probably better data for this species than any other species in the Caribbean in terms of actually the temporal and spatial patterns of spawning. And then, of course, as I was mentioning, we need to know something about, once you've got eggs that hatch and larvae go into the water column, we need to know something about larval behavior. We need to know about planktonic larval duration, about ontogenetic vertical migration, mortality rates, how post-larvae might be attracted. And so we did a number of studies on this. I'll talk about a few of these to help populate this model with actively swimming larvae that move in an appropriate manner. Now one of the ways we tried to figure out where these larvae were at different stages in their life was to actually go back to an archive data set that Bob Cowan had. He'd been working, doing some billfish larval work, in fact. But of course, when he pulled these nets back and forth from Miami to Bimini once a month at 17 stations, a bunch of different depths, it wasn't just billfish getting into those nets, there's lots of lobster larvae. And so we actually were able to come up with looking at some size-specific distributions of larvae from these field samples that Bob had. We also then work with some colleagues in Japan to not only rear the lobsters, because our Japanese colleagues were just masters at doing the aquaculture of these organisms. So we're able to rear it for its first time through all its different stages, 20-something different stages for more than 6 months. But what we did is, as they're aging too, we put them through a battery of tests where we could expose the larvae to different light wavelengths and intensities that mimic different depths in the sea. So we could also try to get some feel about ontogenetic different responses to light. So we could kind of, again, pinpoint where they might be in the water column at different depths. And just to give you some feel for that, these are larval movements, so 0 here would be, this is moving towards the surface of the tank where the light was versus away. And what we found, no matter what light intensities we're working with, is a very clear situation where after about 3 months, the animals before that were positively phototactic, but the key movement was after about 3 months' time, these animals became negatively phototactic and moved away from the light. And the size at which they did that matched very well the size at which we were seeing changes in the field distribution. So we had both field and laboratory data telling us that there was a strong shift at about 3 months in terms of their depth of distribution. The other thing we're interested in is at the final post-larval stage, the little critter that's sitting there on the end of my finger here, that's a stage that doesn't feed, that swims toward shore, it metamorphoses out of the shelf break and somehow senses and moves on shore. But one thing we didn't know is just how far offshore can they sense a coastline. For the model, we need to know at what point in time when you have a larvae moving in a model, what point in time are they essentially captured in a coastal zone in the model. Now we knew that they were attracted to certain species of algae, the chemical senses of different algae. And actually we did a series of experiments then to ask the question, well, how far offshore can they sense coastal cues? Now one of the things we knew from prior studies is that they don't like reef water. There's a lot of fishes. There's a gauntlet of mouths they have to pass by when they come over coral reefs. And so if you test, this shows how many post larvae in basically a choice chamber. What these are on the bottom there, you see these sort of choice chambers. We'd give them a choice, these post larvae, of 1 or 2 water masses. The control was always water that we collected 50 kilometers offshore, so oceanic water. And then we test them against water that was from Florida Bay, the reef, and different distances offshore. And what we can clearly see is that they certainly love coastal water. Bay waters, out 15 kilometers, there's strong selection, 20 kilometers, they're definitely choosing it. They don't like the reef, but about 30 kilometers, we certainly see a decline in their choice. So we chose for our model using about a 20-kilometer sensory kind of capture window. So when post larvae get within about 20 kilometers in our model, we consider them to have settled in that coastal area. And so just to kind of recap then, putting all that biology together, here's what the larvae in the model work on. So the females spawn at the reef edge. For the first 3 months, these animals are vertically migrating daily in the upper mixed layer up down to about, say, 50 meters or so. After about 3 months in the model, they move deeper and are vertically migrating daily down to about 150 meters or so. And at the shelf break, out to about 20 kilometers, that's when the post larvae are able to detect a coastal cue and move on shore. And so this is how we set up all that behavior in that model. Now, one of the other things about this model that had been developed by the folks there at Miami is that although it had been very well validated, the physical oceanography had been well validated, no one had ever been able to say whether this model has really predicting the arrival of fish larvae very well. And this model has been published a couple times in Science. It was, but no one ever validated it. Well, one of the nice things too about lobsters is that we were able to collect some empirical data and test whether it was making predictions of larval arrival that were anything close to reality. So we used post-larval collectors, like you can see over here. We had a couple of sites in Florida, a couple of sites in the Yucatan, where we were able to gather 4 years of data, monthly data on the arrival of post larvae at these sites. And we'd use the model then to actually predict at those particular sites what was the pattern of arrival of the post larvae. Now, remember now the predictions we're talking about predicting using this model where you're spawning lobsters throughout the Caribbean, have them in the water column vertically migrating up and down for 6-7 months, and now we're predicting what's the abundance of those things where these collectors we set up into an 8-kilometer polygon. How good is the plots? So this shows for the 2 sites in Mexico, 2 sites in Florida. In red is what we were predicting, and black was the observed. Not perfect, but given the circumstances, we thought it was pretty, the patterns, the seasonal patterns showed up pretty well, we think, in terms of matching after 6 to 9 months in the plankton, being able to predict the arrival of host larvae into an 8-kilometer area in the entire Caribbean. We thought this was a pretty good correspondence. And it's the first time anyone ever tried to model, to match this. So clearly there's some other things we can do to tune it up even further, but as a first approximation, we thought it was a pretty good match. So of course we can use this model to start asking some of those questions about where do larvae go to, where do they come from, and one of the important things to know, of course, if you're around the Caribbean, is what's the relative magnitude of self-recruitment in an area. Now you wouldn't expect an animal that's in the plankton for 6 to 9 months to have much self-recruitment, but because of the complex oceanography and this vertical migration, it's possible, and certain areas definitely show that. So the colors don't mean anything here, they just help to distinguish different regions, but the size of those circles indicates areas where you see strong degree of self-recruitment, and the small dots are areas that tend to not have much. You can also ask questions about which are the important sources and sinks for larvae around the Caribbean. And this, of course, has some pretty important management implications. So again, the size of the bubble is how much a site is a source or sink, a net importer or net exporter of larvae into the population. And some places are more important, clearly, than others in terms of sources of larvae for the other places in the Caribbean. And of course, using network analysis, one can also look at things like betweenness centrality, which is an idea of sort of intermediate nodes. Are there places around the Caribbean that are important places where if larvae move from one place, there's the population, there are some populations that are sort of important way stations, so to speak. And so, for example, off the coast of Nicaragua looks as a one place where we have lots of connections from lots of different populations. And so those are populations that are going to be really crucial in terms of being able to maintain network connectivity in the Caribbean. And so, once again, from a management point of view, one can say, hey, places off the Nicaraguan shelf are really crucial for maintenance of the sustainability of the species like this. And of course, the other thing one can do is use, if you've ever looked at these connectivity matrices, you can actually try to make predictions using these from the model to say what was the settlement location and what was the spawning location of things. So on the y-axis here are spawning locations. These are different places in the Caribbean. And then on the x-axis is where they end up settling. And so if you look on the diagonal and the colors, the reds mean you have a high amount of settlement. So if you look along the diagonals, these are places that have a high degree of self-recruitment. So these are places that, of course, have more or less their own destiny in terms of management in mind. So the Bahamas, because of the complex bathymetry there, is a place that oceanographically really retains a lot of these larvae. And so management in the Bahamas will have big impacts on the Bahamas. And the same thing for places like the Gulf of Panama. Other places, for example, like Florida, do not. Florida is dependent upon, in a great degree, they have some self-recruitment, as I'll show you, but there's a lot of larvae coming in from Florida from other places around the Caribbean. So if one looks at the Florida, for example, and when you want to talk about some of the implications in terms of international management, Florida, in terms of what it contributes to the Caribbean, is very little. Most of the larvae in Florida are swept into the North Atlantic. Perhaps some of them supply Bermuda, maybe some of the Cape Verde Islands. A lot of it probably becomes DOC in the North Atlantic. But Florida has some contribution to the Bahamas and to Cuba. Florida, believe it or not, we were surprised, but southern Florida, near the Dry Tortugas, has a fair amount of self-recruitment only in the lower Keys. So even for a species that's got a 6-month PLD. But a lot of Florida's recruitment comes from elsewhere in the Caribbean, which means, of course, folks in the U.S. are going to have to cooperate with Caribbean nations, and one can specify which nations that are most important to Florida stocks if we have some confidence in a model like this. So we touched upon some of these things in a recent paper from some of this modeling, but one of the things we think that these models can now do is allow us a much more targeted approach in terms of thinking about international management. we don't have to go just from this parochial system where every country is managing their own stocks as if they're isolated. We don't have to go to the other extreme and say, let's just make the same rules and regulations everywhere. There are certain places around the Caribbean where we really need to focus our attention that are much more important source areas, for example, or much more important internodal links in the network that we really need to be. Those are the focus points we need to. We can't save lobsters or their fisheries everywhere, but there are certain places that are really crucial. And we actually threw out the idea of maybe even thinking about something on the order of larval credits, much like carbon credits, if one is thinking about how to connect different countries and what the actual currency ought to be. We also looked at some things about, I haven't talked about it here, but we looked at MPA designs and whether how one cites MPAs thinking about connectivity in the Caribbean. And then one last thing I'll just show you here in a second is that is the consideration of pelagic MPAs. We really hadn't thought much about this, but we got this idea that because we can, with a model, you can take each larvae, post larvae, that arrives in a location. So every larvae that's settled, we can back calculate, well, where did it spend most of its time while it was in the plankton? Are there any patterns to that? So highlighted in red here, that red means places where the larvae had strong recruitment success, where most of those that finally recruited on shore, where do they spend most of their time in the plankton? And so the areas in red, which turn out to be actually pelagic in some cases, but sometimes places coastal areas, even for this long-lived PLD, this species has a long PLD, the red areas really show where they spend most of their time in the plankton. So this gives us some thought. If these patterns, we repeat this with other species, other fishes, for example, maybe it's a situation if we overlay all those layers, maybe there's certain areas that are particularly important for larval survival and success. And maybe those are some areas we ought to be considering in terms of maybe some special consideration for protection, for example. If you actually follow the light blue areas kind of through the Caribbean, it actually maps very well with a strong current that runs, the major current that runs through the Caribbean, and we refer to this as the highway to hell, because if you're a larvae that gets into that spot, that fast-moving current that eventually becomes the Florida current and the Gulf Stream, those larvae have the lowest probability of actually arriving back to shore and recruiting. So that's a place they really don't want to end up if they don't want to become that dissolved organic carbon. So, pestilence. Usually related to disease. I've had a few graduate students who are sort of pesty from the time I've been. Pestilence has to do with disease. Now, we humans, when we think of disease, we tend to think of it in one of two ways. One, we think of these massive epidemics like the Black Death that have major consequences for what goes on. I think we're all reminded of that with the Ebola scare that's going on. But we forget, of course, there are a number of these diseases that are not always lethal. A number of childhood diseases nowadays are not lethal, and people take their kids to daycare so they get chicken pox or measles or mumps or what have you to make certain of that. And this much is the same way in the natural world. There certainly are dramatic examples where diseases can wipe out and have populations, these major epizootics occur. But most animal populations as well exist in a situation where disease is there in a sublethal effect or only affecting a small part of the population. And all these organisms, for example, all have disease. But they're just not these massive die-offs that we tend to see. So, very similar to what we see in human populations. Similarly, we humans have lots of ways to curtail the spread of disease. And Mother Nature has her ways as well. Symbiotic cleaning stations are great examples of the removal of, in this case, parasites from fishes. There are other ways that Mother Nature has for dealing with diseased individuals too. Predatory culling of the weak and disabled, thank God it doesn't happen with humans, is something that's very, very common in lots of situations and is becoming increasingly documented even in a lot of marine systems where diseased animals, there's higher rates of predation on them, even in cases where the pathogen isn't spread by that mechanism. Now, we humans have another way that we deal with disease. this idea of quarantining. People, another way to call it is social aversion, or if you're a psychologist, you might even think of it as disgust. And there are psychological groups that talk about disgust as an adaptive behavior for disease avoidance. All they're talking about is a social mechanism. It's quarantining. It's a social quarantining situation. And it turns out that there are a few animals that do this as well. It turns out that lobsters are disgusted by diseased lobsters. Healthy lobsters are disgusted by them. They avoid them. And we discovered just a few years ago an instance of this with this host parasite or pathogen interaction between the Caribbean spiny lobster and the PAD1 virus. Now, lest you think that diseases are not important in crustaceans, you'd be wrong. There are a number of various diseases of crustaceans that are of commercial importance, and there have been instances where whole fisheries have collapsed due to disease. So this issue with this virus in spiny lobster that we discovered a few years really concerned a lot of people for a number of reasons. One is that it was the first virus that's been described for any species of lobster, and we were concerned initially whether it was an emerging, potentially epidemic or epizootic kind of situation. It turns out not, but it's highly lethal to, it's lethal, highly lethal, so that's not exactly, it's lethal to 90% or more of the juveniles. Interestingly, the adults don't show that they're asymptomatic. They carry the virus, but we don't see any evidence that the adults are transmitting it. So here's a virus that's definitely lethal for the juveniles. We estimate in Florida that probably at least a quarter of the recruits are killed by this thing. So this is not a small matter. And some places we go, we have hotspots where the disease can be, juveniles, it could be 70–80% of the disease. And that means they're all dead. We don't get juveniles to survive this disease when we get it. Now, it's transmitted, interestingly enough, through contact. Remember, this is a social species. So much like the common cold that's transmitted when you guys are in close quarters with one another, this disease is similarly transmitted through close contact. Also, a little bit in water, but over only very short distances. So this is really a virus that only really works in a social context. It's unlikely to be a human health risk, but like most things, it's difficult to tell sometimes whether these things might eventually evolve and jump. The photos on the bottom there showing healthy and infected just shows, if you look on the right when you're infected, you can notice that if you flex the cephalothorax and the tail, you'll notice in that window there where you're looking at the cardiosinus there, their blood turns this milky white color when they're heavily infected. And that's one of the ways you can do it diagnostically. You can look at it in a late stage infection. We've developed a PCR technique, of course, that allows us to more accurately tell when an animal's infected or not. Now, this virus occurs throughout the Caribbean. We've done some now looks around the Caribbean, and you can see the white dots where it does not occur. The colored dots are various degrees of this. Now, these are estimates based on adults because it was easiest for us to get adults. At each one of these places, we had at least 100, sometimes a couple hundred adult lobsters. Juveniles, remember, it's much more prevalent in juveniles. So the prevalence of this disease in the Caribbean is likely to be much, much higher than this. But the cool thing from the respect of this idea about sort of the analogy with human quarantining is that we discovered a few years ago that healthy lobsters can detect chemically and avoid diseased lobsters. And the real question then is that in a normal circumstance like this, when you have a bunch of juvenile lobsters that hang out around small coral heads or sponges or something like that, how effective is this aversion, this social aversion, in sort of keeping in check the potential spread of this disease? So we know what occurs. We know they don't like to hang out next to another diseased lobster. But how effective or how important as an adaptation is this in nature? Well, it turns out the occurrence of what we might refer as a natural experiment allowed us to look at this question at a very large scale. In the Florida Keys, where we do a lot of our work, there was a massive cyanobacteria blooms that occur from time to time. And in 2007, there was one of these blooms. And one of the consequences of these blooms is that they absolutely wipe out sponges in large areas. So in Central Florida Keys, for example, large sponges, and some of these sponges, loggerhead sponges can be the size of truck tires. And these are important in shelters for lobsters. Sponges are the most abundant animal biomass in Florida Bay in these nursery habitats, and they're the most important shelter for lobsters. And so before and after one of these cyanobacteria blooms, the entire sponge community is in the center of this area, and it's not a small area, roughly 25 miles by 25 miles. The entire sponge community of 40-some species of sponges completely wiped out. Gone. All of them gone. And so one of the real questions is now we've got a shelter limitation issue. Now all the remaining lobsters have all got to be packed in together into those same few shelters. And of course the expectation would be that that disease, the incidence of PAD1 disease, might increase. Did it? Well, first thing, let me just document the changes in shelter patterns. This is a coral head that normally would have 1 or 2 lobsters under it. Corals were not affected by this sponge die-off, of course, they're not sponges. But now, with the loss of all the sponges, the few remaining shelters look like this. Lobsters were jam-packed under these few remaining shelters like this. And one can imagine then, if you've got pathogenic disease, it might just run rampant under these kinds of close quarters. Well, what we see is that definitely shelter patterns had changed. So this is a quick graph that shows that in yellow this is the frequency with which one finds 1, 2, 3, 4, what have you, number of lobsters on the x-axis. So typically shelters have, they often find 1 lobster, but 2 or 3. Blue is what happens at sites these are the impacted sites. You can see that the tail end of that distribution, there's a lot more cases where we're finding 7, 8, 9, 10 lobsters all jammed in together than we saw at places that were unaffected by this bloom. Similarly, if you just follow like a couple of sites, the site in yellow is a site that never had a sponge die-off. And you can see here's where the sponge die-off is tiny. So this is the mean number of lobsters per den over time. Of course, it didn't change where the site just outside the area where the plankton blooms occurred. But you can see there's an example in blue where a site that was inside the bloom, right after the sponge die-off, lobsters again, the density of lobsters per den went through the roof. So, did the virus increase in the population? Well, using 3 different metrics, we're able to determine that it didn't. So this is just the prevalence of this virus on the y-axis, using either that visible diagnostic mechanism, using histology, or using a PCR method. And it's both before and after the sponge die-off at a series of sites. And you can see that after the die-off, which is in the white, there's no spike in the amount of this disease. So despite the fact that lobsters now are all jammed in together, we know it's a pathogenic disease, transmitted by contact, not going up in abundance. So what's going on? Well, is it this behavior? Well, one of the ways we tackle this is to, again, turn to some modeling. We know enough about the life history of lobsters that we've put together. We have an agent-based model. It's a spatially explicit agent-based model. I'm not going to go through the details, but it includes a lot of changes in water quality in the model and how that affects different aspects of lobster life history. But we added the dynamics that we understand about how disease affects these organisms. And this model plays out on a spatial landscape like this, and that are Florida Keys, with each one of these little habitats where we actually specify, based on real data, the abundance of different sponges. And what we basically do is run this big lobster population model in this spatial landscape with and without a sponge die-off. The plot on the top shows in black a time series of lobster abundance. In red is the time series of the disease. Top is where no sponge die-off occurs, and the bottom is where this sponge die-off occurs. Now the patterns look fairly similar until you look on the actual, you look at the scales of the axes. And if you look at the numbers on the top, what you see is when a sponge die-off occurs, we actually see that there's actually about a 50% decline in the number of lobsters in this region. They emigrate from the area, and many of them are killed. The model also shows, I'm not showing it here, that they do tend to aggregate. So the model was replicating what we're really seeing in the field. And in addition, the model was showing that after the sponge die-off, the prevalence of the virus doesn't increase in the model, as long as we keep disease dynamics and this avoidance behavior in play. If you remove the avoidance behavior from the model, the disease spikes, it skyrockets. All right, so what we know about the animal seems to suggest that maybe that avoidance behavior works. Maybe it's adaptive. But we said, let's do an experiment on this too. So what we did was we went out there to these coral heads, like you see at the top again, where all these lobsters were all jammed up in there. And we did a typical kind of tethering experiment. Dauphin Island is famous for tethering experiments and all those and all the controversies therein. So in this case, we tether either a healthy or diseased lobster to a brick and gently swim up and just slip that healthy or diseased lobster in amongst these other lobsters. Now we counted all the lobsters around those coral heads, and we just asked the simple question: is there any difference in terms of 24 hours later, in terms of the number of lobsters we see in those areas? So this is the percent of the dens where the abundance of lobsters either decreased or increased or no change. So whenever you go in there and monkey around with lobsters or any critters, some of them leave. So on the left you can see the white is when we slip the healthy lobster in, and we still get about 50% of the lobsters leave. And there's also just a lot of natural movement in and out of those dens. But when you put a diseased lobster in with these others, you can see we get almost 100% of them leave. It's 90-something%. And you never see, almost never, you see where it says increase or no change. You never see lobsters coming in once you put a diseased lobster. So we think this is even additional evidence. Right in the field, you slip a diseased lobster into these other healthy lobsters, even when they're all jammed in, there's no other shelter around, they leave. It's like being caught between the devil and the deep blue sea, right, in this situation. We've actually done experiments in the lab where we give a lobster a choice between entering a den with a diseased lobster or entering the den where there's a chemical scent of its arch enemy, an octopus. Lobsters hate octopuses. They're Satan to lobsters. About 90% of the time, they'll choose the octopus. They'll choose the quick and rapid death rather than the slow, lingering death of disease. So it's amazing, we think then, this social aversion, this disgust towards diseased lobsters, has evolved in this social species as a very effective way to retard the spread of diseases in this system. And so, much like human quarantining, there are some animals out there that seem to do similar things. Which leads us then to the last point I want to talk about, is alien encounters. Now, we humans have some experience with aliens, here on Earth. And when aliens arrive, it's almost never a good thing, right? For humans, almost never a good. There's always death. There's always destruction. They send down probes and spirit away humans all the time. And it's the same thing for lobsters. They're wandering around the seafloor. These aliens drop these probes down and whisk away their brethren. And Lord only knows where they end up, after they're whisked away by these aliens. And so these aliens have some pretty important impacts on lobster populations these days. This is just a plot showing a fished and unfished population of lobsters, males on the top, females on the bottom. I think it's pretty clear with lobsters, like it is a lot of other species, that not only is there a big decrease in abundance, but it's a very clear situation where it's the large individuals, of course, that are removed from populations. We all know this. Now, for many species, lobsters included, when you have sexual dimorphism in size, it's oftentimes one sex is impacted by this more than the other. In the case of lobsters, males can get about 6 times bigger in biomass than can females. And so in a lobster population that's heavily fished, it is the males that are impacted more than the females. Both sizes are reduced for both, but the males are impacted. As you can see, the males tend to get much bigger, and that's in carapace length. Now, what happens when we lose lobsters in ecosystems in terms of abundance? There's lots of evidence in temperate areas, in fact, that lobsters are one of those species that really can create trophic cascades. Now, the best evidence for this really comes from rocky shore, subtidal rocky shores, and from kelp communities, not so much from tropical species. There's also some interesting evidence that lobster populations can control the abundance of things like urchins, which also can control disease dynamics in urchins. So there's some important ecological things that happen when you lose the abundance of lobsters, but one of the important things when you reduce lobster size is the loss of fecundity and therefore spawning stock biomass for fisheries. So lobsters have indeterminate growth, and they have what is typical of a lot of finfish species, that is an exponential relationship of fecundity, number of eggs per female. And what this graph is not showing, this is a single clutch showing brood size with female carapace length. Big lobsters also will have multiple clutches per year, small lobsters don't. So this relationship is really even greater if you calculate annual spawning stocks. But one of the things we find with lobsters that's very common with all decapod crustaceans is that focusing on changes in female size is only part, in fact, maybe even a fairly small part of the story. Males make a difference. Sperm ain't cheap for lobsters. In fact, decapod crustaceans, I mean, you may think you've perfected sex, but you don't have anything over lobsters. They have perfected it. And how do you define perfection as sex? Sperm to egg ratio. I mean, if sex is really for fertilization success, the best species out there are the ones that produce the fewest sperm per egg. I mean, if you had it perfect, you produce 1 to 1. Decapod crustacean sperm to egg ratios are on the order of about 20 to 1. Remarkably low sperm to egg ratios. Sperm is not cheap. They go through elaborate mating behaviors, and they couple, and they go morphologically have great adaptations to sort of perfect this sex thing. So, as it turns out, large males also in these populations have a much bigger effect on the sexual mating systems. Big males can mate with more females, and they can mate with larger females. Small males actually can't produce enough sperm to fertilize the eggs of a big female. These males will produce many, many spermatophores. We've actually shown in experiments that large males can ration the sperm they give to females. They assess female size somehow, and will transfer a large spermatophore to a big female and a smaller spermatophore to a small female. You don't do that if sperm's cheap, right? You do that because this is a limiting resource in these systems. And so one of the things we also find is that in an unfished population, lobsters display these complex lek-like mating systems, much like an elephant seal does, where the males will take an area, defend it, females come into those habitats, they mate and leave, so males defend territories. But in a fish population, it's like Animal House. It's a mad scramble. Every male and female are running around, mating with one another. The lek system's completely broken down. They don't have any habitat. So unfortunately, most populations around the world of lobsters are so heavily fished, there's almost none of these unfished populations around where the natural mating systems are intact. And one of the things we're really concerned about is the impacts this is having on sustainability in fisheries in the long term. So, in terms of thinking about long-term sustainability of the lobster spawning stocks, one of the things, going back to the connectivity study that we've been recommending and thinking about, is that we really want to focus on rebuilding lobster spawning stocks in these key source populations, right, the blue populations. Forget about the red ones. We can focus now, if we believe that modeling and connectivity story, we can focus our efforts on a few key places, the important nodes and important source populations. We also want to make sure that when we're rebuilding healthy spawning stocks, that's not just big females we want in the population. We know now that we really need big males in these populations as well. And so I think we need to come up with a new way to do this. A new option to consider to help rebuild spawning stocks at large scales. Now, one might say, oh, we'll just throw a bunch of MPAs out there, no-take reserves. Well, I think the reality is they work with lobsters, by the way. I mean, you can build up lobster spawning stocks, big animals in a no-take reserve, but many of them leave too. But the reality is with MPAs, they're too small and too few to be able to really produce enough spawning stock to be able to really rebuild stocks at a large scale. We'll see. I'll show you some of the results that suggest that's the case. Now, another thing we've been thinking about that they use in freshwater fisheries a lot, some coastal fisheries, but they don't use it, in many commercial fisheries as maximum size limits. Now, lobsters are not subject to barotrauma like a lot of fin fishes, so this actually potentially could work. But the problem with maximum size limit, just imposing that traditional kind of fishery management technique on lobsters, is that in the heavily capitalized, overfished populations we have right now, very few lobsters are ever going to get through that gauntlet. It'll take forever to try to build. You can set up a maximum size limit, but the fishing is just too heavy. But if we put these two things together, our sort of thoughts on this is that now you could permit big animals to grow up in those MPAs, and when they leave that boundary, when they're spillover, now they're protected outside. And so this may be a way for us to kind of extend the footprint of MPAs. Now the real question is, how big the MPAs, how effective is it going to be on different fishing intensities, what's the maximum size? There's a lot of details that need to be worked out. But the basic idea, again, is like this. In an overfished population, you got a lot of little lobsters running around, and none of them ever get a chance to get big. Now, if you make a no-take reserve, you can get lobsters in there. They're going to grow big in those reserves, but the no-take reserve by itself, again, as soon as a lobster moves outside the reserve, fishing is so intense it's going to be annihilated. And we're not going to be able to build up a lot of population except within the no-take reserves. If you just have maximum size limits, a fishing size slot limit, we already have minimum sizes. Certainly lobsters might grow big, but again, if they grow big and there's no, they're probably never going to get big enough before they're fished out until they reach that maximum size limit. So our thought again is, if we have both of these, that the big animals that are growing up in these main these reserves, the few that move outside, now they're protected. So now we can build up spawning stocks not only just within MPAs, but maybe we can go ahead and build up spawning stocks outside. So this is not, this is not rocket science, but it's amazing that very few, of any places, is this ever done. I don't know about fin fishes. There's no place in the world that they do this for lobsters. No place. So we've done some modeling work, and we published some preliminary results on this a couple of years ago in coral reefs. We built this very simple stage-based model and asked that question. And what we're plotting here is the number of eggs spawned over the simulation year. And the blue and green lines are the slot limit alone is green, blue is slot limits plus marine reserves. The two lines on the bottom are the current fisheries and reserves alone. And this is based upon some data in the Florida Keys, life history data of lobsters in this situation. So the point here being is that this was a situation where we only had 1% of the population of the area was in marine protected areas. What we're doing now is exploring this approach in a much more detailed metapopulation model, where we're varying MPA area, we're varying fishing intensity, and we're varying different slot limits to see if we can't come up with some scenarios that make sense in terms of mixing this ecosystem-based approach, MPA, with the traditional fishery management approach of maximum size limits, and can perhaps rebuild spawning stocks that have been so overly fished in the Caribbean. And so hopefully it can restore this balance between the sustainability of lobster stocks and then the catch of, for the fisheries that are so important in this region. So just the take-home messages today then. Remember that homelessness is a regular part of lobster's life cycle. Has something to do with dispersal and connectivity, and I think we're learning a lot about that and its importance to management decisions. Pestilence is also a regular part, everyday life of lobsters and a lot of other species, and in this case, I think we're just on the verge of discovering this idea of social aversion, its importance in retarding disease in species. We haven't really looked at it too much in a lot of other species, but I suspect it's much more common out there in the natural world than we think. And finally, in terms of alien encounters, I think it's no clue, no new news to you that we have big impacts on these populations. But I do think we have to start looking at maybe some new ways to potentially build up spawning stocks. For lobsters, I think that's the biggest crucial issue in the Caribbean, is rebuilding spawning stocks. And maybe the one way we talked about is an approach that might prove useful. So thanks for the opportunity to come talk to you guys here at Dauphin Island, and if you have time, I'll have to answer a question too. Thanks.
We've got time for a couple questions.
Yeah.
Mark, in your biophysical modeling, you predict these source regions, and then you have sinks as well. And if you look at the incidence of the virus, it seems like it maps pretty well with the sink populations. Have you guys explored that?
Yeah, we have actually. We have a paper in press right now in ICES that's coming out where we looked at these sorts of things. We were concerned about the virus. We actually found the virus in post-larval lobsters, and we're actually wondering whether it might move around with larvae. And we looked at it in more detail and didn't find any evidence of that. But we did a modeling study using the same model to look at disease transmission using different planktonic PLDs of disease. And the short answer, prone to long answers, the short answer to your question is, yeah, it does seem to map pretty well some of those certain areas in the Caribbean. The northern Caribbean seem to be disease hotspots, and there's some real disconnect from some of the places in the Eastern Caribbean and the Southern Caribbean. And when you start looking at some of the network nodes, there's something going on still with this transport of this virus we don't understand. If you look at the nodes, make sense. The connections make sense over short time periods. So you can't model it. They're definitely not in a 9-month time period. But if you model it with a 1-week time period of disease transmission, it maps pretty well to some of these populations that we're seeing. What you're saying, some of the source populations. So I think we need to do some more of that. But there's definitely some disease connectivity, we think, at small scales like that. So, yeah, there's no question about it.
I know they're not built for it the way that American lobster is, but when they're molting, is there any incidence of cannibalism? I mean, they're a social creature, and it looks like they need shelters together. But what is
Yeah, that's a good question. In captivity, they definitely, they'll eat anything, of course. So when you hold them in captivity, if one molts, they do. Actually, in nature, what they tend to do in the social, when they get ready to molt, they'll move away from them. So the recently molted animals are oftentimes by themselves, so that there's not cannibalism in nature. So they're not naturally cannibalistic, but if they were forced together, the others would eat them. Yeah.
That's an interesting problem to find shelter if you're already using shelter in the hard shell form and in clusters, and now you have to find shelter by yourself.
Right, and we've never looked to see, it's interesting, are those animals then perhaps taking subpar shelters, as they're getting ready to molt perhaps? And I've actually never looked at that, to be honest. One of the other concerns we have is that when you use, I'm going to talk about this, but some of the artificial shelters that you use around the Caribbean, particularly things called casitas, these little small artificial shelters that fishermen use rather than traps, there's some good and bad aspects. We have a paper coming out on that referred to as Ecological Traps. One of the issues is that when you put these casitas in nursery habitats where there's a lot of molting, you're concentrating a lot of small animals that normally are by themselves, and now they're all being forced together, and we were wondering, does that affect growth rates, does it affect disease, does it affect mortality? And what we're seeing in there is that you do force a lot more animals together. We're seeing higher mortality. It may be part due to this more frequent molting. The other issue is that these small animals, since they could detect this chemical scent of a big lobster, a lot of small animals that are normally by themselves are all being drawn to those casitas. And as anybody who works with artificial shelters, it's not lobsters that are only attracted to those things. There's oftentimes a lot of big predators around. So we think these casitas are probably where you have adults that are not molting very frequently and are used to being together, big, and hanging out beyond the gate limits of a lot of these predators. Casitas are probably a fine fishing mechanism for those populations, but if you put these things in nursery habitats, it looks like they have a negative impact on juveniles because of being drawn to the predators and more frequent molting.
With the larval transport in those nodes off of Nicaragua, is that purely driven by circulation, or is that something that if they're going to be moving around the area, this is a stopover spot to eat and persist?
Yeah, well, we're thinking that it's definitely a lot of this connectivity or the self-recruitment driven by circulation issues, of course, and it's an interaction between behavior, because as I've shown you earlier on, if you don't have behavior, you get very different predictions about where they go. What I mean by that kind of case, of course, is that I think those are important intermediate populations in terms of connections. And so if, for example, a situation like Nicaragua, if you overfish that area or the habitat in that area is degraded, then that normal connections that would normally between, even if you think about demographically or genetically, between, say, South America and Florida, you're going to disentangle those. So now animals that maybe one generation later could have come from South America are no longer going to be able to make it through that node. That node would basically destroy that link. And we got to be careful places that are important to have multiple links. We destroy habitat or overfish those areas, we've disconnected that bigger network. And I think that's the real key thing in terms of looking at internodal fishing. Yes.
One last one.
Can their prey, like the octopus, can they differentiate between the diseased lobsters?
No, in fact, what we see is evidence of that predatory culling. So when the lobsters are diseased, we've actually seen this in blue crabs, and we've done some studies with hematodinium too. We just published a paper recently on that. In both of those cases, when the animals are diseased, it's the classic scenario that predators are much more effective in preying upon diseased animals. And unlike some parasitic diseases where you have an intermediate host, things like hematodinium, the dinoflagellate disease that affects a lot of crustaceans and these viruses, they don't need an intermediate host. So a predation event really is a culling mechanism. It helps keep the spread of disease, contained. So, well, thanks again for having me.
All right.
We have a lab, I don't know. Well, it's like it's a separate going away for classes. So I don't.
I think what we're going to do, I would start sending it to you could show up, send budgets for the whole.